We are Choi Song-myung from Korea and Yoon Jung-won from Korea, who researched on the utilization of waste decomposition by microorganisms in the intestine of insect larvae. Recently, because of COVID-19, using of disposable products has been multiplied. The decomposition period of highly polymerized compounds, the main ingredient of disposable products, takes about 100 to 500 years, so it affects the ecosystem a lot. Also, highly polymerized compounds flow into ocean, causing a large number of marine organisms death. To solve this problem, we studied how decomposed polymer compounds using intestinal microorganisms of insect larvae. It is known that our sample of neurons and some larvae can be composed using microorganisms in the body used as food and release inorganic waste and carbon dioxide form. What they have in common is that microorganisms that can break down highly polymerized compounds such as cellulose and wax live in the intestine. Based on this theoretical background, we additionally selected silkworms and caterpillars because they eat similar foods and have high possibility of decomposing macromolecular compounds and research. We have installed breeding boxes suitable for its selected larva's habitat and supplied styrofoam to certain sites for neurons which confront plastic tables. The intestinal microorganisms of larvae were instructed to proceed with the first culture. Because we don't know what kind of environment microorganisms survive, we proceed in both aerobic and anaerobic environments. Experiments conducted in an aerobic environment were conducted on a clean bench, and intestinal microorganisms were extracted from its larva's intestine and incubated in LCA2B made of the following combination. Because the medium does not contain carbon source to feed microorganisms, samples of plastic, styrofoam, and paper fragments were added. Therefore, microorganisms cultured in this medium can be determined to have polymer compound resolution. In the case of anaerobic conditions, the experiment method is the same, but the experiment was conducted and incubated in anaerobic laboratory. After 4 weeks, the incubation results of the medium made in its environment were confirmed by measuring S1. When measuring S1 was measured using its control group as a reference, and as a result it was confirmed that microorganisms were not cultivated in the aerobic environment and that microorganisms were cultivated in lava silkworm and caterpillars in the anaerobic environment. Therefore, in the subsequent experiment, we decided to experiment in anaerobic environment with only the superworm, mealworm, and waxworm. Formerly of the first culture in anaerobic conditions were breached in a new LCA2BM medium produced in the same way as before. During the first incubation process, we made 2 bottles of superworm and mealworm and 1 bottle of waxworm. In addition, we made a CFBM solid medium to check culture aspect and isolate microorganisms. Solid batches are made 2 bottles for each larva. 4 weeks later, we measured the absorbance and found that vacuum and superworm had absorbance measures that could be used, but mealworms were difficult to use. On the other hand, CFPM confirmed that colony was well formed in all lava. Colony was formed near plastic and the other two types of lava were formed only near styrofoam. Therefore, we confirmed that there was an error in the absorbance measurement process, and we continued to experiment with three types of lava. In order to check the cellulose resolution of microorganisms when conducting the third system culture on LCFDM, microbial culture was also carried out on CNC media. Normally, on secondary culture was extracted and spread on its CNC medium and incubated in an anaerobic chamber. Also, we wanted to check the degree of decomposition through mass measurement of samples that were placed in the first culture 8 weeks ago. As a result of incubation, large quantities of colonies were identified in the superworm medium and colonies were identified in the mealworm medium. On the other hand, we could see that the waxworm did not form a colony due to characteristics of lava that digest protein. Intestinal microorganisms are interpreted to have excellent cellulose resolution, and other experiments such as absorbance are expected to show good results because these abilities are related to breaking down plastic and artificial polymer compounds.
The research of measuring the mass change of samples by recovering all samples from the primer salon where samples were stored for 8 weeks are as follows. In the case of styrofoam, mealworms had the highest rate of change, except for silkworms and caterpillars, which had been lost during the sample recovery phase. In the case of PAT, mealworms had the highest rate of change, and in the case of sterile paper discs, waxworm had the highest rate of change. Particularly measured, the mass loss rate of silkworms and caterpillars in styrofoam was the largest, but the loss rate seems to be unusually large compared to the integer absorbance measurement because the serum had the small opening. Mealworms and waxworms also had massive loss rates in PET samples, indicating that the intestinal microorganisms of the two larvae had broken down PET. In the second culture serum, samples were recovered and attached to the surface of the sample to identify the growing microorganisms, and we observed it with a SAM. We observed the surface of styrofoam, plastering, and PET bottles, all of which had a smooth surface in the control group, while the surface of the sample was uneven in mealworms, and superworms had a wide distribution of many microorganisms. This concludes that robust intentional microbes successfully attach to the surface and decompose the specimen. It was also found that the results of the secondary absorbance measurement process were errors because it was confirmed that microorganisms were successfully in attaching and inhabiting samples in mealworms culture. To determine the type of quantity of gases released during microbial proliferation, we used DCMS measurements. Gas was collected from other secondary culture serums and injected into DCMS to measure two types of carbon dioxide and methane. As a result of measurements, methane was not detected in all four serums. This is expected to be due to the absence of methane-producing bacteria among microorganisms. On the other hand, carbon dioxide was measured 10 times higher than the control group in all serums. Most of the existing studies conducted incubation in anaerobic environment and measured the resolution accordingly. But the effect was minimal or showed little. Therefore, we conducted the experiment in an anaerobic laboratory. As a result, unlike previous student studies, we found that intentional microbes were cultivated under anaerobic conditions and concluded that some of the intentional microbes could use plastic as the carbon source. In addition, CNC culture to confirm cellulose resolution showed that intentional microbial colonies were formed closely related to plastic and cellulose resolution. The scanning electron microscope confirmed that microorganisms are attached and inhabiting artificial polymer compounds provided as carbon sources. Furthermore, the GCMS track showed the microorganisms were actively decomposing compounds, judging from the high carbon dioxide concentration. So far, research has been to confirm the presence of microorganisms that are likely to degrade styrofoam and plastic, and to confirm the ability of microorganisms with actual resolution. Currently, waste such as styrofoam and plastic are supplied as the only carbon source in the search culture serum and seed feeding medium to multiply for a long time. However, there was disadvantage that it was difficult to measure accurate ability and efficiency because there was a natural amount of compound falling out of the water. Therefore, we are planning to measure resolution of efficiency by measuring the concentration of water dissolved in culture by filtering even small pieces completely. We will also sequence and isolate after mass incubation the determined species composition. Later, we will produce a model equipped with microplastic filters to efficiently cultivate plastic and styrofoam pieces that are small and floating in the culture. The references are as you see now. Thank you for listening.